Now we're going to focus in more on one of the eukaryotic cells, the animal cell. Okay, so one version of eukaryotic cells. So this is the type of cell that you have in your body. Okay, this is just the general kind of form of that. So this is looking into an animal cell and looking at the different organelles that you'll find. So we're going to go through each of these, okay? Some we won't go into like super specific, like the actin filaments and the intermediate mediate filaments, okay? But we are going to go over the majority of this, okay? So first up, the cytoskeleton. So the cytoskeleton is basically, as you might guess from the name, kind of the backbone of the cell. So that is kind of the greenish image, or the greenish part of this image here, okay? It supports and shapes the cell, so it kind of holds the sides in and basically offers kind of the support structure inside the cell so that the cell doesn't just kind of wobble apart, okay? It also helps to position and transport organelles. So it is sometimes referred to as the highway system within the cells. Okay, so the different organelles can kind of attach onto the cytoskeleton and get moved around the cell wherever they need to be. It also provides strength for the cell, okay, because the cell can move by kind of stretching out and then coming back together and stretching out, coming back together. And so during all of that, it can be kind of stressful on the cell. So the cytoskeleton really kind of absorbs a lot of that and provides the strength of the cell itself. It also assists in cell division. So again, by moving organelles around, it separates them out so that there's one of each type of organelle on both sides of the cell at least, and then also kind of helps pinch the cell together to complete that cell division. And then it also aids in cell movement, which I already kind of talked about. Okay, so moving on into the nucleus. So the nucleus stores the genetic information. Now, like I said in the previous section, it does still have to be open to the rest of the cell so that the DNA and the genetic information can be read. Okay, so that's where you get these nuclear pores. Okay, so they're little holes in the nucleus, in the membrane of the nucleus, the nuclear envelope, and they allow for passage in and out of RNA, okay, and then some, ribosomes, which we'll get into later, and then also sometimes some proteins, okay. So the nucleus contains the nucleolus, okay, so that's the denser core that you see right here, okay. The nucleolus is where another organelle called the ribosomes are made. Now we'll get into the ribosomes more later. They are incredibly important to the cell. So we'll definitely talk about them quite a bit. Okay, so these are the organelles involved in making proteins. Okay, so the nucleus stores the genetic information, and then that genetic information can be copied and then taken out into the cell. Generally, it will go to the ribosomes or the endoplasmic reticulum. Okay, I know that's really kind of a weird word, but endoplasmic reticulum, often shorted as ER. Okay, now there are two different types of endoplasmic reticulum. Okay, you have the smooth endoplasmic reticulum, obviously has smooth sides to it. Okay, and then you also have the rough endoplasmic reticulum, which is studded with these little dots. Now these little dots are called ribosomes. Okay, now ribosomes are incredibly important in making proteins because they actually link the amino acids together. Okay, so the ribosomes take the genetic information from the RNA that made a copy of it on the DNA, and then they start actually reading it and putting it in a usable format of the amino acids. Okay, so this is where you get that organic molecule being produced. And if you remember from that chapter, chapter 2, okay, when we talked about amino acids and proteins, proteins really help carry out most of the chemical reactions inside the cell. So if you didn't have proteins, if you didn't have ribosomes, the cell basically wouldn't be able to do anything. Okay, so those are the little dots that you see all over this. There are a ton of ribosomes in this image alone. Okay, then you also have vesicles. So vesicles are membrane-bound sacs that hold materials. Okay, they're kind of like the little storage bubbles inside the cell. So you can have tons of vesicles. Okay, they can move nutrients around the cell. They can actually be produced by taking nutrients into the cell. They can also push waste products out of the cell. Okay, you also have the Golgi apparatus. So the Golgi apparatus is known as kind of the UPS system of the cell. So it's a bunch of like stretched out vesicles, basically. So there are these long kind of loops of membranes that process the proteins that come. They also sort them out and then ships them off where they need to go. Okay, so you'll notice that there are a bunch of vesicles associated with the Golgi apparatus. So basically these proteins get shipped into the Golgi apparatus in these vesicles. The vesicles will actually fuse with the membrane of the Golgi apparatus, drop their proteins off, proteins get processed, they get sorted as they continuously go from one membrane to the next through vesicles, and then they eventually get shipped out in these little vesicles at the end. Okay, so there's a lot of motion and fusing of membranes and all different kinds of things happening in the Golgi apparatus in order to, create that processing, sorting, and shipping network. All right, and then probably the most famous, if you've been online anytime in the past couple decades, is the mitochondria. Mitochondria produce the energy for the cell. They are actually kind of a weird organelle. They have two membranes, so they have your inner and outer membrane. Yeah, we'll get into that more later, but there is one other type of organelle that we'll see in the next section when we talk about plants that also has a second membrane, okay? And it is very useful for its function. You also have vacuoles. So vacuoles, you're generally going to see them more prominently in plant cells, okay? So this is actually an image from a plant cell that shows you a massive vacuole. Vacuoles are fluid-filled sacs that hold materials. So this is where the cell can store nutrients, it can store water. Plant cells will actually use this one massive vacuole to create the rigidity in the cell. Okay, so it fills it up with water so that the cell kind of sticks up and gets stiffer, okay, so that it can hold its shape. All right, so if you have a plant and you're watering it frequently, okay, it's going to stand upright. Okay, the stem is going to stand upright. But if you go away on vacation for a week and you come back, well, that plant has used up the water that it had. Okay, so it used up all the water that it had in all of these different vacuoles and all the different cells. And so now, because it doesn't have that filled vacuole, it droops. Okay, so basically plants stand up using water. Kind of interesting little fact. You also have lysosomes. So lysosomes, if you think about Lysol, okay, I always connect the two in my head. Lysosomes come around to digest material. So they help keep the cell healthy. Okay, they will eat different waste products. If there's an organelle that is kind of failing in its purpose or it's worn out or anything like that, the lysosome will basically come up and fuse its membrane with that other organelle and then just kind of eat it. Okay, so they are kind of the garbage disposal of the cell, right? And they have a lot of enzymes inside the lysozyme that actually helps with that digestion. Okay, so if a lysozyme bursts inside the cell, that cell is pretty much going to die because it's releasing all of that digestive material that's usually contained inside the lysosome, but it's releasing all of that into the rest of the cell. So a lysosome bursting would be a really bad thing for a cell. All right, and then finally we have the centrioles. Okay, some people call them centrioles, and I don't care. It's one or the other. So centrioles are tubes that are found in the centrosome. Okay, so you've got your centrosome here, okay, and then the centrioles. So they are tube-like structures. They mostly help divide DNA. There's still a lot of questions about what centrioles really are, what they do, all of this kind of stuff that we don't really know all that much, because some scientists actually took centrioles out of a cell and then watched to see how well it divided, because the whole hypothesis was that they wouldn't be able to divide very well, and yet they divided just fine. So we really don't know what they're for, but centrioles form cilia and flagella. So if you remember back in, I think it was the previous section, I showed you that cell that looks like a pepper, okay, like you might find in a Papa John's box, okay, and it had, I said, this long string kind of thing basically worked as a propeller to move it. That's a flagella, okay. Cilia are little hairs that grow out of the cell membrane and kind of wiggle back and forth, almost like a centipede, in order to move the cell, okay. All right, so we're actually going to go back to the picture that has all of these. Okay, so we talked about our cytoskeleton, supports the cell, helps move things in and around the cell, and plays a role in the motion of the cell itself. And we also talked about the centrioles, the complex assemble of microtubules, so they are connected in with the cytoskeleton, these tubes of proteins, things like that, that help divide DNA. And we talked about the cytoplasm in the last section, okay, the kind of gel-like fluid that kind of contains all of the organelles and keeps them a little bit separated. We talked about the mitochondria, where you get metabolism basically taking place to create usable energy for the cell. The lysozymes, which are the digestive components of the cell. Okay, we talked about the Golgi apparatus, which gets the proteins, collects all of the proteins in, and then starts to process them and ship them out around the cell where they're needed. We talked about the membrane a little bit, the cell membrane, a little bit ago, like in a previous chapter. Okay, so you've got your lipid bilayer, the phospholipids that we talked about in chapter 2, okay, that form kind of a double-layered membrane, and that surrounds the cell. We didn't talk about the peroxisomes, okay, they are kind of sort of related to the lysozymes, but don't worry about that. We did talk about the ribosomes, and we're going to get into those more in depth in the next couple chapters. So they are small complexes of RNA and proteins that are the site of protein synthesis. And there are two different types of ribosomes. There are free ribosomes, which you'll see just kind of floating out in the cell. So we've got a couple down here. Okay, and then you also have the bound ribosomes that are part of the rough endoplasmic reticulum. We talked about the nucleus and the nucleolus. So the nucleus is the command center of the cell where the DNA is contained and the RNA is produced. And we also talked about the nucleolus where the ribosomes are produced. Talked about the nuclear envelope, which is another membrane surrounding the nucleus and separating it from the cytoplasm. And the nuclear pores, which are the little holes in the nuclear envelope. And then we talked about the smooth and the rough ER. Okay, smooth is really used to manufacture carbohydrates and lipids, whereas rough endoplasmic reticulum is used to create proteins, used with their interactions with the ribosomes to create proteins. Okay, so the information goes from the nucleus into the smooth or rough ER, and then through the ribosomes to produce the proteins into a vesicle, which then goes to the Golgi apparatus or the Golgi complex, and then out to the rest of the cell.